and welcome to Brewing Brilliance, a podcast where we talk to academics from across the university who are involved with the Healthy Ageing and Chronic Conditions Research Institute. We will be speaking to researchers who are leading innovative interdisciplinary approaches to promote lifelong health, delivering improved interventions to manage chronic conditions more efficiently. These sessions are essentially opportunities for us within the Healthy Ageing and Chronic Conditions Research Institute to meet colleagues from across and beyond our faculty to foster knowledge sharing, collaboration and networking. My name is Adrienne Cleverly, and today we are joined by Dr. Martin Gill. Martin is a senior lecturer in chemistry whose research is at the interface of chemistry and biology. This interdisciplinary approach aims to isolate novel molecules suitable for applications in areas such as drug discovery and cancer research. So welcome and thank you for joining us.
Thank you very much for inviting me.
So that's quite an unusual background there in terms of the chemistry and biology. So can you just give me a brief overview of your background and how you got into the two?
Yeah, sure. So I'm trained as a chemist, so I did an undergraduate in chemistry, and then I wanted to do research, but I wanted a chemistry project, but I wanted to really have an application in mind. So rather than sort of pure chemistry, I really wanted to make molecules with some quite nice purpose about them, if you will. And within that, I ended up doing a PhD, which was awesome fun, which was basically a mix of the two. So I did a bit of synthetic chemistry, characterizing molecules and so on, but then also quite a bit of cell culture and microscopy. So I got to look at the molecules I made in cells, sometimes killing all the cells, sometimes not. But then also they were actually visible under light microscopy, so I could actually see where they were in the cells, which I thought was really cool. So that really sparked my interest in this interdisciplinary approach. And from that point on, I always wanted to know one bit more. I always wanted to know what more can they do in cells, or what other molecules can I make and what can they do? And so then that really just led to my fascination with this mix of the two disciplines. And yeah, my whole research has been still based around that concept, really.
You've got a lot of experience then working across the two disciplinaries. So have you got any different branches that you think work well, or barriers? What do you think across the two?
So I do think it's very fun, first of all. So I really like having that experience of both fields and working with scientists, both in chemistry and biology and even medicine. So you get exposed to loads of different ways of thinking. We can't know everything, so it's very useful to work with people who know things you don't, and I really think that's something very important in research. And so in some ways, one of the initial challenges was probably just slightly different ways of thinking about research and different ultimate aims as well. So a chemist wants to look at their own molecules, of course, whereas for a biologist, they don't really mind what molecules they're using, as long as they're getting the inhibition of their protein or the thing they want imaged. And so it's been fun and occasionally a bit challenging trying to find out where I can fit within these different aspects within the different disciplines. So that's something which I certainly have taken a lot from it.
Do you find that there's not necessarily language barriers, but using different... Well, you laugh then. So
well, sometimes, yeah.
Yeah, across the two. Because when you've got the two different branches and they come together, how do they connect?
So I think it used to be a lot more siloed. So I think early on when I started, it did feel as though chemistry departments, biology departments were quite separate. But I think there's been a big push in actually getting researchers to speak to each other, present work, sharing PhD students and things like that. So I think those connections have really evolved as a result of that. So certainly maybe the next generation of scientists coming through are then a lot more naturally interdisciplinary in how they work because of those different exposures. I mean, in terms of how they naturally connect, as I was saying, I think it did used to be a bit more siloed, and it would be a case of biologists working on their problems, and then we need inhibitors. We've got a chemistry group we can work with. We'll ask them to do it. The chemistry group does it, passes it on to the biologists, who then carry on. So I think there was a bit more of a different team approach and then passing on the results to each other. A bit like how a pharmaceutical company would work, in fact. Whereas I think more recently people are actually really talking with each other very much at the start of projects, and really chemists are trying to say, right, what do you guys want? What are you trying to achieve? And then from very early doors, you incorporate these aims, these ways of working into your own experimental designs. So I think that's definitely improving over time.
And I like the way you said that it's evolved, because immediately when you started talking about it, I thought of the same thing, because lots more researchers are having or trying to sort of embed themselves in that interdisciplinary practice. So to hear that you're living and breathing it and you've got really positive experience of it is just
Yeah, absolutely.
what we're
And
hoping for, I
yeah,
guess.
yeah, and it's very fast moving in the sense that, of course, computational power has just increased enormously within the last, well, I mean, in my time, so in the last 20 years. And so computational chemistry is really big, as you'd imagine, not to mention computer science as a whole.
Yeah.
And you can see how that is spread everywhere. Everyone wants to get on board with it. It can be a bit difficult if you're not particularly computer savvy or computer literate
Yeah.
to think about, oh, I've never coded before. But there's a whole massive department in Swansea, for
Yeah.
example, of people who code all the time and have since they were very early in their careers. And you just want to chat to them and say, well, we've got this problem. We actually don't know how it fits with what you're doing at all. And then you'll just have these interesting parallel conversations. So
And just grow.
yeah, exactly, and it can just grow. And I think that's so important for the evolution of science. Is yes, you've got your own specific topic and aims, and we wouldn't be researchers if we weren't a bit crazily self-obsessed with something which maybe others don't think is that important. As in, that's kind of part of our jobs and our definitions. But I think to have that open-mindedness and to be willing just to think, oh yeah, actually, I wonder if I could be applying this for a different purpose, or I wonder if there's other techniques, other concepts, methodologies from other emerging disciplines which could be applied in mine. And so I think that's kind of what makes it fun, really, for me.
So that broad sort of thinking of could this, how does this,
Yeah.
how do you choose what molecules you want to study?
Oh well, yeah. I mean, there's so many of them. So
Yeah.
I have an undergraduate lecture where I actually ask the students, how many molecules do you think exist, and how many could exist theoretically? And the answer is such a big number. It's more atoms than will ever exist in the universe. So you have potentially almost quite literally an infinite number of molecules. So yeah, how does a chemist decide, I want to work with this one or
Yeah.
these small section? And it's pretty random reasons, really. So there was just stuff I did as an undergrad. So I really enjoyed transition metal chemistries. I liked the d orbitals, for example. I just thought they looked cool. and then that interest just grew, and I just started to really enjoy the structures the molecules made, the types of bonding interactions, then their actual properties. So a lot of these molecules have really nice emissive properties. So a lot of them are luminescent, which is why I did all that microscopy on them. And these properties are quite distinct to this area of chemistry. So it just became self-reinforcing, really, and I've enjoyed working with them for quite a long time now, sort of 20 years or so.
It's interesting as well, because you touched on a little bit there about like how they sort of react and things like that. Does that sort of help you decide like how the applications move into sort of the health and side of things? Was that sort of a progressive? Because
Yeah, so a lot of this work is based on the work of others as well. So I
Yeah.
think it should always be acknowledged. You are taking a small step from, off the backs of numerous other researchers who've done a lot of the work as well. So yeah, in terms of the properties of these molecules, they had several advantageous aspects, which were then we thought, I mean, my supervisor at the time, and then I've continued some of these ideas further
Yeah.
forward, but we thought they'd be really good candidates, in this case, initially for cell imaging because of these luminescent properties. And then as time has gone on, I've realized, well, a lot of their properties actually make them really, I think, intriguing candidates as potential therapeutics. even just from the point of view that they're quite different to a lot of conventional therapeutics. And so even a very simplistic way of thinking that if you have unusual molecules, you can have unusual things happen in cells. And if you're lucky or careful or work with great biologists, then you can often try to expand that, find out more, and then you can maybe have some interesting potential therapeutics on your hands. So that's kind of been how my work and approach has evolved over the years.
Have you had any unexpected outcomes?
Oh yeah, all the time. So I mean, I think the very start of my PhD, my PhD supervisor basically said, make some molecules, chuck them on cells, and see what happens. And with that in mind, everything that's happened has been unexpected. But in particular, we had a really nice cell imaging molecule. So this molecule was really good at imaging DNA. So you could treat cells with it, and you'd see a really nice nuclear staining. So a classical DNA imaging agent. Very pretty images. We loved it because it was a good application of our molecules. And then we continued looking at it, and we realized, as well as imaging DNA, the molecule was also stopping cells proliferating. And all of what we assumed turned out to be absolute rubbish. So we thought, because it's imaging and DNA and binding DNA, the mechanism must be something to do with DNA. For all the experiments I did, all of them were null results. And this is where working with biologists came in very useful, because we had everything carefully controlled, which took me ages to learn and master, in honesty.
Yeah.
All of the controls were absolutely fine. The molecule was doing nothing to DNA at all. It turns out that molecule was actually inhibiting actin polymerization. So actin being part of the cytoskeleton, so responsible for the shape of cells. If you inhibit actin, you can then inhibit cells moving, spreading on a surface, but you can also inhibit cells dividing. And this molecule was inhibiting actin polymerizing, and as a result of that, the final stage of cell division, mitosis, was blocked. And there was no way we ever would have predicted this would happen. From a DNA binding molecule, DNA imaging in cells, to an actin polymerization inhibitor. Absolutely unexpected.
And it just sort of sounds out as something that you remember, because that didn't come from just chuck it on and see what happens, though.
Well, it sort of did, because I was doing some live cell microscopy, and we had a bunch of cells under the microscope, and we imaged them for quite a while. And then I remember bringing in my PI, who was then a biologist at that time. I remember bringing him in and saying, Carl, have a look at this. What's going on with this cell? And of course, I hadn't much experience of this, so I didn't really understand what I was seeing.
Yeah.
Carl, being a very experienced and talented cell biologist, he said straight away, there is something going wrong with mitosis. And from that, we were then able to work backwards to actually figure out what was going on, which did take about 10 years or so. But
wow. But that's just crazy that, you've got that result from that, but also you've highlighted again the powers of interdisciplinary research.
Exactly.
Because you as the chemist and him as the biologist, and you just, wow. So how might advancements in medicinal or chemistry disciplines contribute to managing chronic diseases such as cancer?
Yeah, I mean, that's a really interesting question. So I think this is led by the medics and the biologists. And I think we've seen, I mean, genetics has just really come into its own over the last 20 years or so. And this has then promoted this concept of personalized medicine. So if you think about what we can actually do with a patient who has been diagnosed with a disease, we can actually now look at their genetic code and then make a prediction on what molecules, what drugs, what treatment they would be the most likely to have success within that patient. And this can be done on a patient-by-patient basis, which is, I'd say, it's limited by cost at the moment, but we're not far away from this being the case. And so within this, I think if you have more drugs available, which can then be tailored, but I think from a medicinal chemistry point of view, we can certainly be looking closely at our molecules to fit within this personalized treatment aspect. So being well aware that it's not going to be a one drug fits all. Even if pharmaceutical companies want that, I suppose. But if we have multiple derivatives of a molecule, you can actually potentially fine tailor a drug for a particular patient. And in terms of synthetic chemistry, I mean, I'm probably doing myself out of a job here, but with the advancement in automation, you can actually feasibly have benchtop synthesizers for molecules now. So there's nothing, I see it as perfectly feasible in the fairly near future to have basically a bedside pharmacy, if you will.
Yeah, I was going to
and
say,
labs can use things like
it
that.
sounds like it's becoming much more person-centred in
Yeah,
terms of
absolutely.
it's literally the person at the centre, not necessarily the
Yes.
drug.
Yeah, exactly. And you know, there's other things within med chem which are certainly still have a big contribution from chemical scientists. So fields such as protein inhibitors, things like that. I mean, we saw with COVID, I mean, the speed at which we needed to develop treatments for that. You had, the key aspect was having an RNA, a nucleic acid based vaccine, and that somehow needed to be protected from degradation. That's where chemists come into play. So you can either make modified nucleic acids, which aren't degraded until they're in the cell and do what they can, or you can have protective capsules, so sort of within a drug delivery type approach. So this is still where I think chemists will find a lot of application as well. So I think you've kind of got loads of different areas where we're still seeing quite big advances.
See, every time I sit with somebody opposite me, I'm just like, wow, at what they're talking about. I genuinely love research because it's endless, isn't it?
Yeah.
But opportunities, I mean. I'm going to put you on the spot with either the easiest question or the hardest
okay.
question. What's your favourite molecule?
What's my favorite molecule?
Yeah.
So, if I had to choose one molecule,
Definitely put you on the spot.
you have put me on the spot. So I obviously can't choose one of my own. That would be far too egotistical.
You can.
So I think my favorite molecule would probably be something like ferrocene. So ferrocene is one of the classic organometallic molecules within this world. It was discovered in I think it was the early 20th century. So it kind of formed the start of the whole area of chemistry, which I'm interested in, and the molecules I work with. It's got a really unusual structure. So it's known as a sandwich complex.
Yeah.
And so it's got this iron atom sandwiched between these two organic ligands. And so I always love lecturing it because I always love just thinking about sandwiches, maybe. But it's just got this really cool structure. It's a really stable molecule as well. And it's been, like I say, it's been known for ages, studied for ages. And the interesting thing is it doesn't actually have many applications. But despite its own humble, maybe lack of applications, it's very stable and has actually sparked interest in a whole area of chemistry, which I still follow to this day. So I'll choose the humble ferrocene.
I take that as a good answer from you. Thank you. I've got so much more I could ask you, but unfortunately we've run out of time.
Yeah, no problem.
But thank you so much for joining us and talking about your experiences in research. If you would like to find out more about what Martin spoke about, you can visit him on his staff profile pages at Swansea University, or you connect with the Gill group.
Yeah. Feel free. Yeah, feel free
and
to
see
email.
what projects they're involved with. You can also get in touch with us at the Healthy Aging and Chronic Conditions Research Institute. Thank you so much for joining us. I really enjoyed that.
Thank you.